So we are at part 3 of the notes for viruses. And just as we do in class, I'm going to go ahead and do a quick recap. So we talked about that we're learning about viruses. That's what these notes are on. Definitely get your notebook out. These are the objectives that you should be making sure you understand and can answer the questions to or explain. These are your vocabulary: virus, capsid, lytic infection, bacteriophage, lysogenic infection, prophage, and retrovirus. We talked about the discovery of viruses a long time ago, and we also talked about the virus structure and composition, meaning what does it look like, how is it made, and virus reproduction, saying that it's non-living and it can only reproduce by infecting living cells. We talked about how a viral infection is going to a host cell is similar to an infection going into a computer or a virus going into a computer. We talked about the two types of viral infections, which are lytic and lysogenic. And then we went through the process of a typical lytic infection. Step 1. Step 2. So step 1 was the virus injects DNA into a bacterium. 2 was viral genes are transcribed by the host. 3, the bacterium makes new viral proteins and nucleic acids. Step 4 was the proteins and nucleic acids assemble into new viruses. And step 5, they bust out. All right, the viral enzymes lyse the bacterium cell wall, and the new viruses escape. And that leaves us to here, where we're starting with this one, lysogenic infections. So, step 1, the virus injects its DNA into the bacterium. The viral nucleic acid is inserted into the host cell's DNA, where it is replicated along with the host DNA without damaging the host. The virus's DNA, it actually becomes embedded in the host DNA and is called a prophage. For the prophage, the viral DNA again inserts itself into the bacterial chromosome. The prophage may remain part of the DNA of the host cell for many generations of reproduction or replication. It is replicated along with the host DNA without damaging the host. Viral DNA multiplies as the host cell reproduces. So, the viral DNA, when you multiply something, you get more. As the host cell reproduces, the viral DNA reproduces. In this way, each generation of daughter cells derived from the original host cell is infected. We know that daughter cells come from parent cells. So, we know that those cells are also going to be infected. The prophage may replicate with the bacterium for many generations. So, this should remind you of mitosis. Remember MIT, make it two cells? So, we see right here, there's one parent cell, and now it's making 2 separate bacterium for quote-unquote future generations. Right? So again, the prophage may remain part of the DNA of the host cell for many generations. It is replicated along with the host DNA without damaging the host, and the viral DNA multiplies as the host cell reproduces. Each generation of daughter cells derived or coming from the original host cell will be infected because of this. Next, the prophage can exit the bacterial chromosome and enter a lytic cycle. So, from a lysogenic infection and going to prophage, it can go into a lytic cycle. So, let's kind of talk about this. Influences from the environment, such as radiation, heat, and certain chemicals, they can trigger the prophage to become active. It then removes itself from the host cell DNA and reproduces by forming new virus particles. The lysogenic infection, at this point, becomes an active lytic infection. Now, so for extra credit, yeah, I hid it inside the video. For extra credit, write this down and you can take a picture and send it to me. You can send me a voice clip on Remind app of what you feel the answer might be. But for extra credit, my question for you is I want you to think of an outlaw, somebody that is running away from the law in the Wild Wild West, like way back in the day, right? And I want you to kind of compare it, use an analogy to compare it to a lysogenic infection. So, my question for you is how would you modify the story of the outlaw in the Wild Wild West to make it analogous to a lysogenic infection, to a lysogenic cycle? If you have any questions about it or you need help, please let me know. Contact me. If we can have a conversation about it so that I know that you understand, you're good. Points done. Everything is Gucci. Okay? It's all good. So, as long as I know you understand what we're talking about, I'm good. If you have no idea and don't understand, still call. Because what does that show me? That shows me that you are taking initiative to find out. And I'm all about that. All right? So let's go ahead and move on to the next slide. An RNA virus, the common cold. So once the cold virus has penetrated the host cells, it uses the host cellular machinery to replicate itself. So it uses what the host has to replicate. About 70% of viruses contain RNA rather than DNA. In humans, RNA viruses cause a wide range of infections, from relatively mild colds to influenza or the flu, and also AIDS. Certain kinds of cancer also begin with an infection by viral RNA. Crazy, right? So, what happens when you get a cold? Simple. The cold viruses attack with a very simple, fast-acting infection. That means that it is going to come in and immediately start replicating. Understand that a capsid settles on a cell, typically on the host nose, and it is brought inside where a viral protein makes many new copies of the viral RNA. So, the virus makes many copies, again, of its RNA. Now, the host cell's ribosomes, and we remember that ribosomes is an organelle, which means little organs, and they are inside cells, and ribosomes make proteins. Okay? They are the protein factory. So, the host cell's ribosomes make the viral RNA for the host's own mRNA, and it translated into capsids and other viral proteins. So it takes what it has and it's changing it and translating it into something else. So, what did I just say? The copies are translated by the host into new viral parts. That's another way to say it. Next part. The new capsids assemble or form around the viral RNA copies, and within 8 hours, the host cell releases hundreds of new virus particles to infect other cells. What did I just say? Right? The parts assemble into new viruses and burst from the host cell. Just that simple. All right, next part. A retrovirus. When we're talking about a retrovirus, when you hear the word retro, you always mean like back in the day, right? It's not technically back in the day as much as it is backwards. So, a retrovirus. The most commonly known one is HIV. In contrast to the cold virus, HIV makes a DNA copy of itself that inserts into host DNA. There, it may remain inactive for many cell cycles. So, this deadly disease let's talk about this deadly disease called acquired immune deficiency syndrome, which is also known as AIDS. It is caused by an RNA virus called human immunodeficiency virus. So HIV causes the syndrome of AIDS. HIV belongs to a group of RNA viruses that are called retroviruses. The genetic information of a retrovirus is copied from RNA to DNA instead of DNA to RNA. When you think of the statement that we were saying in class, DNA makes RNA, makes proteins. If this is copying it from RNA to DNA, but we know that DNA makes RNA, makes proteins, something is wrong. That's backwards. And that's literally what retro means. It's going backwards. It flipped. So, in the next video, we're going to go through the process of an HIV infection, giving us an example of a retrovirus.